Good morning, good afternoon, and good evening, everyone, and welcome to this Inside Scientific webinar sponsored by Iconius. This is Liam Sanyo from Inside Scientific, and I'll be your host for today's event, which is titled Functional Ultrasound Neuroimaging in Awake and Behaving Non-Human Primates. Today, we're being joined by Dr. Pierre Pouget and Dr. Serge Picaud, who will provide an overview of functional ultrasound imaging and how it can be used to study the brain in behaving non-human primates. And with that, I'm very pleased to hand things over to Dr. Pierre Pouget. Pierre, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thank you. It's a very big pleasure to show you some of our recent data acquisition in the lab. And you may be familiar with ultrasound imaging or not, but let me show you a few slides. What we mean by ultrasound imaging is the use of a transducer array made of a piezoelectric element to construct an image. The way it works very briefly is an electric pulse is transmitted to a transducer that will transfer this electric pulse to ultrasound pulse. The pulse will be reflecting by the surface that is imaged, and the echo pulse that will be received by the transducer will be transferred back to an electrical signal to generate an image of the tissue. This is a brief overview of two images made of the brain of rodent. On the left side, you could see a conventional Doppler that has been used for a few years, but in the late of the 2000 and in the year of 2010, the group of Michael Tanter made a big jump by generating ultra-fast Doppler imaging. And as you can see on the right, the main interest of this new image is that the signal-to-noise ratio is much higher, and you can see very more detail of the vascular tissue. This has some important consequences, is that we can have more sensitivity, and then we can record cerebral blood flow and blood volume changes during modification. Here it's an image of a coronal view of the brain of a macaque, and I will go back to this in a few slides. And this was a low determination of functional hyperemia, so change according to task or according to behavior of the animal. So the way we study these cerebral blood flow changes in macaque is to use a regular chamber. So here what you could see is a regular cylinder chamber that has been used for many years in electrophysiology. And the probe, the ultrasonic probe that you could see on your left side, is the size that has been designed to enter into this cylinder. So there is no specific modification to electrophysiological setup. So you can see the head post on the right side of the monkey skull. Here is the side view of the monkey's head. And for the talk of today, I will be talking about the medial prefrontal cortex recording that has been made in the lab, and Serge will present some of the data made in the visual cortex. You could also see on the left side that one of the interests of this technical approach is that not only we will have a precise view of the vascular tissue close to the surface of the brain, but we can go deep as 10 millimeters, and we will have a field of view of 14 millimeters, accessing to a deep structure. So the way we do study the brain in macaque in the lab is very common to many labs in electrophysiology. We train monkey to do oculomotor task in the lab. So they are trained to sit in a chair, they receive juice while they are doing simple or complex visual task. And while we record their eye movement, we also record electrophysiological activities such as single cell recording. And we of course do online analysis. So while we did start to study with Alexandre Dizieux in 2019, this question using ultrasound is we were really stuck by the result. And what I will show you is the really first data we record with Alex in the lab in a very simple way, because we didn't know what to expect for at first. What the monkey has to do is to do a simple oculomotor task, in that case Cortex Pro or anti-saccade task. What you will see in this movie is the really first series of data acquisition we got, and I illustrate this by the fact that here what you will see is really no post-processing data. So this is really online data acquisition that we observe while the monkey was doing the task. So here, as you see, the monkey is facing the screen, in a similar way that we do for electrophysiology. And here in that movie, what you will see, this is a fastest movie, but during the first 60 seconds, so 1 minute, the monkey is not doing anything. And on the left side, you could see the change in vascular blood flow. And on the right side, you could see the position of the eye of the animal while we are recording this change in cerebral blood flow. Now, what you could see after 1 minute is the task start, and you could see immediately on the region of the pre-SMA called the supplementary eye field, is you could see a very dramatic change in cerebral blood flow. When I say dramatic change in cerebral blood flow, here on the right side, you have the scale bar, and it could show you that the signal was changing up to 50%. So on each single trial, we were able to detect change in activity of the brain of that macaque. So now, if we start to quantify very simply this acquisition, what we do and what we have done is simply record these activities. So here in red, you could see the change in cerebral blood flow without the rule before the monkey is doing the task, and in green while the monkey starts to do the task. And what you could immediately see is this big jump of change of activity. But then what we could of course do is simply by doing a Fourier transform is to extract the slow brain activity frequency. And of course, we can also extract cardiac pulsatility. On the right side, again, what you could see is a focus on the time at which the task starts. And you could see that each red dot here is a trial. And you can see that even on a single trial, we were able to detect this change in cerebral blood flow. So now to quantify this change, what we have done here on the left side, you could see this focus on the sample rate of change in cerebral blood flow. And what you could see here is that if we take a pixel of a region of interest in the region of supplementary eye field here, that for a large region of interest, so that is almost complete for the full supplementary eye field, is that almost every trial we were capable to detect a change between no rule and rule. So each single trial, from before the start of the trial or after the start of the trial, we were capable to detect that the monkey was doing the task. And in fact, even if we reduced especially this resolution by taking only 1 pixel, so 1 pixel in this region of interest that you could see on the left, 1 pixel was sufficient. Of course, there was some more noise, but 1 pixel was even sometimes sufficient to detect whether the monkey was in condition of waiting for the task or doing the task. And of course, we did a control region that didn't show any significant change while the monkey was doing the task or not. So for us, it was a big surprise because the signal to noise is very, very high, which allow us not to average too many trials, which is very often the case while you do electrophysiology recording or of course, imagery such as fMRI, for example. So now what we wanted to do next is try to see what this signal is telling us about the brain function. And in fact, what we do in the lab is we do single cell electrophysiology in awake monkey. So if you're not familiar with this field, what it means is we introduce a microelectrode in the brain of the monkey, and then we try to be as close as possible to be able to detect spike, which is by definition slow task because you have to be close enough but not too close to be able to record this single cell activity. One of the major issue or problem that we have is if you see these two features on the right is that we are completely blind during this process. So we do not see the microelectrode. So we are listening the brain, we are listening the change in electrical field surrounding the microelectrode, but we do not see the microelectrode per se. So we have to reconstruct where the electrode was and to estimate, for example, in which layer, or to be more specific, in which part of the region we are recording the activity. So now what we did next is try to use this spatial resolution of the ultrasound imaging to localize online the depth and the change position of the microelectrode. So what you could see on the right side here is basically the probe, the imaging probe, while we are dropping the electrode in the brain. And the idea was to try to co-localize the recording of electrical field while we were doing the imaging of the change in cerebral blood flow. So here is a sagittal view of the setup. So you can recognize the probe, that the white part in the center of the screen, and you could see the microelectrode on the right part that is going through the field of view of the probe and allow us to visualize the microelectrode while we are doing the electrophysiology. So this is to show you the very first visualization we have. And here you could see while the electrode was slowly advancing into the brain, and you could see the change in the responses. The electrode is moving, so the imaging, ultrasound imaging is capturing these movements, so you can localize where the electrode is while you are doing your electrophysiological recording. So this allows us to record simultaneously the microscopic recording of fuse. So we were able to record change in cerebral blood flow in such a large region of interest for us, the supplementary eye field. Simultaneously with the local field potential, which is believed to be a more mesoscopic information about what the brain is integrating or what the neuronal network is doing. But also simultaneously, we were able to record single cell and single spike activity. And this is something that we are done now. And this is a paper from the two groups of Serge and myself, and the paper will be submitted soon. So our idea behind this recording would be to, in very much more simplistic way than combining functional imaging and electrophysiology, which is very big challenge due to the electromagnetic perturbation of the signal. Here we did not find so much difficulty. Of course, there was some challenge to fix and orient properly the probe, but then we have been able to record in several sessions simultaneous single unit local field potential and change in cerebral blood flow. And now our idea is to combine this information to understand better what is the contribution to local field potential, single cell activity, and multi-unit activity to change in cerebral blood flow, in particular in a region that are integrating multiple activities such as the supplementary eye field. So one of the, I wanted to finish with this slide because for us working in electrophysiological lab of non-human primate and doing complex tasks, these techniques allow us to not only access large spatial region of interest, but also being able to study this changing activity in a very short temporal and time resolution. So we can really record change in cerebral blood flow, as I show you, in terms of a second and sub-second change in cerebral blood flow. So then we can combine this technique to other technique that has been developed in for electrophysiology, such as multi-unit recording, single unit recording, even optogenetic approach, to be able to better understand what the brain is doing in awake behaving macaque. And again, I want to thank Iconeus for the invitation, and Serge will present some of the common work we have done with his group. And thank you very much, and I will be very happy to respond to your question.
Thank you, Pierre, for the fantastic presentation. And with that, I'll hand things over to Dr. Serge Picaud. Serge, thanks so much for being with us today. The floor is yours whenever you're ready.
Thanks a lot. I'm very happy to present this talk on functional ultrasound neuroimaging, a tool for assessing visual restoration strategies. So indeed, I'm really working on visual restoration, and the idea is that a lot of people are becoming blind, and you see that by 2050, the number of blind people will double, and the people with vision impaired will triple. And this is because of aging with AMD and glaucoma. And the idea is that we want to restore some vision in a blind patient. And so especially we have two diseases, one where the patient are two types of disease, one where the patient are losing the photoreceptors. So, and as you can see on the left, the photoreceptors are disappeared or crossed by a red cross. And you see that you are left with an inner retina with neurons. And the thing is that you have no information in these inner retinal neurons. But can we restore some information in these neurons? And the other type of disease is where you lose retinal ganglion cells, the cells that are sending in visual information to the brain. So if we want to restore vision in these different patients, we can either, when a patient with lost photoreceptors, either activate these inner retinal neurons with a subretinal implant or with optogenetic therapy as indicated by the green spot on the retinal ganglion cells. But this is for the retinal level. But if we want to try to restore vision in patients having lost the retinal ganglion cells in disease like glaucoma or diabetic retinopathy, then we need to directly activate the visual cortex. So we were developing these strategies, and the idea was that we need some way of assessing what is the visual information at the visual cortex for both types of strategies. And so it's why we started to assess whether we could use functional ultrasound imaging to assess this strategy for restoring vision. And so the first was to develop it on rats, because we were not at all a specialist of ultrasound imaging. And so as you can see here, we started with a rat. And we had a cranial window on the skull of the animal so that we could assess directly on the brain and generate some measurements while we were doing some visual stimuli. And you see these were high frequency or low frequency stimulation of the animal with some light presentation. So when we started, we were very happy to see that we could measure activity in the visual cortex, but also in the superior colliculus and in the lateral genic nucleus. And you see these activity. So for the different nuclei, both the visual cortex in blue, in green for the superior colliculus, and LGN in green. And so you see that each stimulation was really generating a significant measure. So, and we could label, in fact, all these structures and see the activity in these different areas. So then, I mean, we were able, by moving the probe, to do a whole skull or whole brain imaging so that we could see the structure in all directions. And this was very interesting. But then the next thing was to really get information. And we wanted to see whether we could localize the information. And this was what we did by doing some local stimulation. You see here, the flicker presentation was either on the right corner on the upper panel, and on the left corner on the lower panel. And you see that the activity was really localized, suggesting that with this technology, we could easily do retinotopy of the brain. So we were very happy with this first result in rats, and it's when we moved, in fact, to non-human primates. So you see here on non-human primates, again, we had to do a craniotomy to have the probe on the jaw. But what you can see already on the upper panel is the image with MRI as compared to first. And you see that the first anatomical image is much better. Then we were having the primate, so he was awake and doing a visual task. He was concentrating on the fixating point in green, and we were presenting some visual stimuli on the side. And what you can see is that we could measure the response either to stimulation with 500 millisecond stimulus presentation or 2 seconds in red stimulus presentation. And what you can see is already with one trial, we were able to detect a change in activity. But the more we were going, and you see that with 10 trials, we had a very clear response after averaging all these measures. And you can see that we have a very nice measurement or localized image on the brain, as you can see here, with both 500 millisecond stimulation or 2 second presentation. And so with this, we decided that, in fact, for the following studies, we would only take the 0.5 second stimulation because it was providing a sufficiently clear measurement and very reproducible measurements. So then, using this strategy where we could localize clearly the activated area corresponding to a visual presentation, what we did was to present different stimuli so that we could generate the retinotopic map of the visual cortex. And you see here that so we try to colorize the visual cortex, primary visual cortex, using, in fact, the different activated area in the retina. And you can see that the macular area was really in the superficial layer of the visual cortex, as expected, and that the more we were going, were deeper layers. And you can see that we did the measurements not only with circular stimulation, but also with angular stimulation. So then we could also do the measurements in different positions, but here it's only the presentation in 2 non-human primates, so on the coronal and sagittal axis. The next question was whether we could really define the resolution of the measurement in non-human primates. And it's where we decided to move to the measurements of ocular dominance column. So it's well known that, in fact, in the visual cortex, using deoxyglucose, it was possible to assign to one eye or to the other some ocular columns, so that when you stimulate with one eye, you will have a predominant stimulation of layers. And if you would flatten down the visual cortex, it's possible to, in fact, have such band representation of one eye in dark and the other eye in white. And so we wondered whether it would be possible, because this has been measured by anatomy. It was also measured at the surface of the visual cortex with optics, but it was not possible really to do measurements deep in the visual cortex. So we wondered whether with the first we could really measure those columns, ocular dominance column. And so when we did this, we could clearly see the ocular dominance column here in the visual cortex, not only in the superficial layer, but also here in the deeper sulcus layer, on the calcarine sulcus here. You see clearly these ocular dominance column, exactly as in the anatomical slice of the visual cortex. And so then what we did was to measure the activity here in different region of interest. And you could see that we were able to see these columns that were clearly designed, and that in fact they were ranging in the correct size according to what was reported, that these columns are between 400 microns to 700 microns. So we were in the correct range, and we could see that we were able to detect them in the superficial layer and in these deeper layers. So this was very interesting. And also, what was quite nice is that if we would try to dissociate or define all the layering of the visual cortex, we could see these ocular columns from layer 3 to layer 5, and sometimes also in layer 6. So then I would like to show you some data that were obtained during our studies of visual restorations. So we have been doing visual restoration with retinal prosthesis, and you can see this retinal prosthesis that is now in patient, and you see how, in fact, these implants are stimulated with an infrared light at the back of this black bar here, and the patients are able to recognize some letters now. And so this was developed in the lab, and we tested on some animals, and you see rats, for instance, here. And we could show that when we stimulate the implant with infrared, we have a clear activation here in the superior colliculus, and also that it was when we were not activating the implant, the light was out of the implant. We had no activity, but of course we could also activate the system when we were having a visible light on the implant or outside of the implant, because this implant is only sensitive to red light. And of course, when we have this, we have a larger area covered. But what was very interesting was that, in fact, we could test different light intensities, and you see that the size of the response is increasing with increasing the intensity. And so we could really confirm that what we had been recording ex vivo on an isolated retina about the threshold of the stimulation was exactly corresponding to what we were measuring with the functional ultrasound imaging. We have been also developing optogenetic therapy, where we use the ionic channel, photosensitive ionic channel, to be expressed through AAV in retinal ganglion cells here. And so this is now in clinical trial, and you can see the first result of a patient who was able to, thanks to the activation of these retinal ganglion cells with an optogenetic therapy. And so you see he's able to find out some object on a table, even a very small object here. Maybe I just wait for that. You see he's finding the object. And so we wondered whether this strategy that was here developed on the retina, whether we could also use it to activate the visual cortex. And you see that we use the same microbial opsin crimson air here expressed in cortical neurons. And you see the cortical neuron expressing this optogenetic protein. And then what we did was to stimulate, and you see that we have neurons responding to the visual stimulation and here to the optogenetic stimulation. The latency is, of course, shorter with optogenetic therapy. And using functional ultrasound imaging, it was very nice because we could really measure the macroscopic area that was stimulated by the light stimulation. And we could also, what was very impressive was that we could have a parallel measurement of the electrophysiology and also functional ultrasound imaging. But what you can see on this slide is that, in fact, we have a similar result, measurement of the activation with electrophysiology, single cell, and functional ultrasound imaging. And you see here that we can really rely on functional ultrasound imaging as well as on electrophysiology. And this was even more clear when we were looking at the transfer of information from the visual cortex after the optogenetic activation to the LGN. Because when you see here, we could record sometimes the activity of neurons, but it was very difficult and we had only very few neurons that were responding to the activation of the visual cortex by optogenetic. But what was very nice is that we could clearly demonstrate by functional ultrasound imaging the activity, and it was much more simple to see, in fact, where the activity was present, whereas with electrophysiology, we were lucky to be sometime in the proper region. So the conclusion that we can draw from these studies is that we can have a nice functional recording of the visual system in rats. The temporal resolution of the activated areas could be correlated. We had retinotopic maps in behaving non-human primates. We could detect the ocular dominance column even in the deep V1 layers. Functional detection of ocular dominance column up to layer 5. The functional response to prosthetic activation of the retina. The functional response to the optogenetic activation of the visual cortex. High correlation with electrophysiology, and so which was quite nice because we could more easily detect the activity in the poorly activated LGN structure. So with this, I would like just to thank all the participants to these studies, and thanks also Iconis, who provided us a research system. We used a system that was a system from Iconis to do all this recording. And also Pierre Pouget, who really helped us a lot for all the study on non-human primates. And of course, Michael Tonter for all this work on ultrasound imaging and some funding. Thank you very much.
Fantastic. Well, thank you, Dr. Picaud, for a fantastic presentation. We're now going to move into the Q&A session. And as a reminder, if you do have a question you'd like to ask, you can submit it using the Ask a Question panel shaking on the screen. All right, Pierre and Serge, all set to answer a few questions?
Yeah.
Sure.
Fantastic. All right, let's start with a question that I think would be relevant to both of you, but Pierre, maybe you can lead us off here. What are the respective advantages of electrophysiology, functional ultrasound, and fMRI?
Well, I mean, the way I see it is, of course, the gold standard is to, record the neuronal activity. I mean, to exactly know what the neurons are doing. But sometimes it's difficult to define the region of interest. So I think FUS is, to me, is a great advantage to localize where the electrode could be inserted and lose more time, and it's much more precise than fMRI. I mean, you can have a couple of dozen or hundred of micron of precision. So that's first. I could see it as an in-between methodology between the two, the coarse response of fMRI and the electrophysiology. The second, I will try to be brief, but the second is it's also a nice method to do an exchange with what is observed in human, because monkey fMRI, of course, it's feasible, but it's a lot of complicated time to train the monkeys, and combining with electrophysiology. I mean, you can do it sequentially, but it's complex. And the last thing, and that's what we are currently doing in the lab, is we can also start to answer, or at least study some of the question about what is the relationship between these two signals. I mean, is there sometimes some differences, some discrepancy between local and more fine signal recorded in the neuron? I mean, are they always correlated? I mean, are they stable over time of a session, for example? And what is the cross-trial variability between this signal? I mean, this is the sort of question I think that is could really be nicely at least studied using a combination of these methods. So, yeah.
Yeah, that's a really great point. And Serge, I'll pass things off to you. In your experience, what do you think are the advantages of electrophysiology, functional ultrasound, and fMRI?
Well, I think with electrophysiology, everybody agrees that you have a high temporal resolution that you cannot get with the functional ultrasound, although you can measure the latency between different areas of how the activity is arriving in different areas. But you will never have such a precise temporal resolution as electrophysiology. But the greatest advantage is finally that what we saw, and I was very surprised, was that we have a high resolution with functional ultrasound imaging. And so it was possible to see all the areas that were activated in the activated zone. And so with this, I mean, it's all, even when we activated the visual cortex and watched to the LGN, I mean, although we had very few neurons that were activated, I mean, with FUS, we had very reproducible results to find out the activity. And it was much more difficult to be in the correct position with the electrode to find out some activity. So I think it's by having a very microscopic view, but very sensitive view using FUS. I mean, it's much faster to demonstrate activation of the area. So, and of course, it's much more simple to have the probe on the brain rather than having the electrodes crossing the brain. So I think it's a great advantage because it's much faster to demonstrate activation of the area.
Yeah, great answer. Another question here. So functional ultrasound has obviously changed quite a bit in the last few years. How has your experience using it changed your view on functional ultrasound? Maybe Serge, I'll get you to lead that one.
Well, yeah, at the beginning, I thought that functional ultrasound imaging would be not highly sensitive. And I thought you would need high activation of an area in order to detect its activity. But I was really surprised when, in fact, having done this work on both with the retinal prosthesis and optogenetic therapy, showing that in fact we had the same threshold of activity to detect the activation of the tissue, of the circuit. So especially, I mean, to show when we were activating, for instance, the visual cortex with optogenetic, that in fact we needed the same amount of light in order to induce really activity in the visual cortex with both FUS and electrophysiology. So I think this really demonstrated that we had a high sensitivity of the FUS, and this I was not expecting that measuring blood flow would be as sensitive than what we'd seen.
Fantastic, great answer. All right, moving on to the next question. What EEG frequency bands are most correlated with the FUS readout? Pierre, maybe you can lead this one.
Well, thank you, Sarah. So maybe we will have to wait for the paper. No, I'm joking. But so this is something we are currently finalizing the clearly. So if you're talking about EEG, we do not record EEG at the moment. So we record the local field potential. I guess it will be really interesting in primate clearly to record EEG and FUS simultaneously. Yeah, so that would be next. I mean, we have done ELFP. There is a clear difference from and stronger correlation between some frequency band that are do not seem. I should be cautious because we are finalizing the analysis now, comparing to fMRI. But as far as EEG, we did not record this signal yet. So, well, I mean, yeah, I mean, if someone wants to come and do it, you're very welcome.
Fantastic. So this sort of leads to the next question. Does the FUS signal correlate better with spikes or for local field potentials? You're saying you were looking at local field potentials. Is it because they correlate better, or what's your
Okay,
experience with
so so now you're trying to scoop the article, so I could not publish it at all because everybody will know. But well, to make the story short, it depends. So first is we see a strong correlation between FUS and spike because we have higher resolution than fMRI. At least that's my interpretation. So compared to what I've been shown is, I think we could say that a spike actually single unit correlate with FUS, but not in a large region of interest, of course. So it's very close to the tip of the electrode. And the second point is that it may be not constant, so it could vary during a session, during the trial. So the contribution on spiket and LFP may vary during the course of a trial. So I hope that's answer to your question. But we can exchange by email if you, because the archive is not out yet. So yeah.
Fantastic. Yeah, great answer. Serge, we'll direct the next question to you. So these measurements of vision are coarse and poorly correlated to fine vision. But what do you think the functional ultrasound signal would look like for finer stimuli, like with higher spatial frequencies? And do you think you can measure visual acuity cutoffs using this method?
Well, this we haven't yet done, and it's clear that we will move on to the more precise measurements. I mean, this was a first study, and we just wanted to see whether we were able to do any measurements, and really, but this is our next step, really, to move into this type of measurements.
Fantastic. Yeah. All right. Next question, Serge, I'll also direct to you. So thanks a lot to both speakers. Very nice presentations. Do you have any idea about the minimal stimulation duration? So Serge, I know you talked about using a stimulus of 0.5 seconds and 2 seconds, but did you use anything shorter than that? Do you have any experiences to share?
Well, when we stimulated with the either optogenetic and also with retinal prosthesis, it was shorter time than this in order to induce response. So I think depending on the stimulation that we are using, I mean, we don't need to have a so long stimulation of the circuit.
Fantastic. All right. Here's another question. So, do you think it would be possible to do this kind of recording at some point with intact scalp? Pierre, maybe you can lead this one.
I hope so. That's why I asked Michael and Icodeus to improve their sequence. Possibly. So I guess this is more a question for the physician, but yes, it might be possible at some point to correct for skull distortion using some lens and other. I mean, I'm not a specialist of ultrasound, but my view is that it might be possible, and there is other option also. We try. We are trying at the moment is to use some contrast signals, so inject some echographic contrast agent to increase the signal to noise and see whether we can get some signal from with the intact skull. This is really too preliminary to say that we could do it, but yeah, I mean, I guess I'm always happy to work on that, of course, to try to get the data from the intact skull.
Mm-hmm. Yeah, definitely an exciting avenue moving forward, if possible. All right, here's an interesting question. So how is the animal's non-local blood flow changes, like blood pressure and overall flow rate, how are those related to body movements reflected in the functional ultrasound signal? So in optical wide field calcium imaging in mice, the animal's body movements can induce a robust blood flow change in the cortex, which can result in false signal changes in optical imaging. So yeah, I guess the question is, how is the animal's non-local blood flow changes related to body movements, and how do those show up in the functional ultrasound signal? Pierre, can you lead that one?
Yeah, I mean, thanks, Keita, for your question. So I guess this is when the question was asked is what I was surprised the most. The first was the signal to noise ratio, and the second one was the way we can deal with sometimes body movement. I mean, there are macaques. I mean, there are big guys, so they can shake the chair, but when it's happened, we can clearly correct for it using some signal processing. But this is very uncommon. I mean, this is no, we clearly just exclude when there is a short shaking or short movement of the body of the animal, and so we can deal with that. Even the pulsatility, because we remove the skull. But we keep the conjunctive tissue. So of course the dura is there, and the conjunctive tissue could be kept, which for me, at least that's my interpretation, allow us to reduce a lot this movement, because pulsatility is very reduced. And the probe is even pushing a little bit on the tissue for the electrophysiologists that are in the audience. So it's helped to reduce the movement. The body movement, we do have sometimes some body movement, but because the signal to noise ratio is so high, we can, whether we just remove the trial, because or we can easily correct for it. So yeah, I know it sounds for me it was a big surprise, but comparing to optical imaging, it's just fine. So and yeah, I mean, and again, I mean, I would be very happy to share movies and data if you're really interested and look by yourself. Yeah.
Fantastic. Yeah, great answer. All right, next question here. How do you envision the clinical translation of these techniques? So Serge, I know you talked a bit about work in humans, but more on the optogenetic therapy, but maybe you can lead this question.
Well, clinically, functional ultrasound has already been used in infants through the fontanelle, and it could also be used during surgeries, brain surgeries, because in this case where you try to locate the part of the brain to remove, you could maybe use functional ultrasound, because usually the techniques that are used are mostly superficial ECOG or some electrodes, penetrating electrodes. But I guess it would be a way to have a view of the whole brain or whole structure. So I think it could be quite useful. I think for infants, it could be also a nice way, because you can repeat the measurements as many times as you want. And it's much easier than MRI, and much more precise. So I think there's great avenues for this technology in clinics, but only for patients where you have removed the skull. There are also some sonolucent materials that could be also used.
Fantastic. Yeah, great answer. There was a question about how early in development can functional ultrasound be used. Would it be appropriate at early postnatal time points? But I guess you addressed that one in the same shot too, so that's great. Serge, do you have any data showing layer-specific activation differences using functional ultrasound imaging in the V1 or elsewhere?
Well, when we measured the activity, I mean the ocular dominance column, we could define the layers and show that we had really activity in some layers and not in the other layers. Like the activity was from layer 2 to layer 5, and it was clear that we could see it. But other than that, we haven't tried to really make some specific layer activity.
Yeah, fantastic. Great answer. I think we'll make this next question the last one. And again, we don't mean to try to get the scoop from you, but maybe we can end by just giving a brief summary of what are the next steps for the Pouget and Picaud lab. Will you merge optogenetics with functional ultrasound and non-human primates, or what do you think is the next step? Pierre, maybe we can start with you.
Yeah, so maybe with this answer, I will try to answer some of the Jeff question at the same time. So I guess for me, one of the question will be to examine the spatial resolution we can get, for example, in functional resolution, such as variability of latency, saccade position in front eye field and supplementary eye field, or even the spatial representation of, for example, saccade control in supplementary eye field, not putting the probe in coronal view, but in sagittal view to cover, the a more rostral caudal representation in supplementary eye field. For the comparison with BOLD, we do not plan to do that, not even simultaneously or on the same animal, but we probably, or actually we do already start to compare some responses between labs in Lyon. So comparing with Sugan Benamet to compare, for example, some physiological response we observe in or two monkeys, so two type of behavior that respond similarly. And so we have the fuse data, she had the fMRI data, and we can compare. It's not completely satisfactory, but it will be a first step, at least on our side.
Fantastic, thanks, Pierre. And Serge, what about you? What's your next steps?
Well, our next step is really to see how objects are represented in the visual cortex, because this is what we want to do for visual restoration, is to try to define how to stimulate the visual cortex in order for a blind patient to see. So we would love to use this technology really to define how the objects are represented in the visual cortex.
Fantastic. Well, thanks so much, Serge, and thanks, Pierre, for sharing your insights today. It's been a real pleasure having you with us.
Thank you very much.
Thank you.
So in closing, thanks again, everyone, for attending this Inside Scientific webinar sponsored by Iconius, and we'll see you again next time. Have a great day, everyone.